I'm Matt, the MCAT boss, and let's chat about what you need to know about protein denaturation. A term refers to the alteration of the native structure of a polypeptide macromolecule, the loss of its tertiary structure, sometimes the loss of its secondary structure, specifically because weak bonds get destabilized. Denaturing is not the same as digestion. We are not separating amino acids and dissolving the protein chain. We are just disrupting the folding pattern. Now, there are a couple of really specific things that can happen to drive this. Extremes of things like temperature or pH, the extreme concentrations of things like salt or solvents or urea or other chemical agents. The most common example is heat. A higher level, in this case, tertiary and quaternary structures are disrupted by the heat. Now, if we plot this on a graph, you can see that as the temperature goes up, the denaturing continues to disrupt that higher order structure. This is what that tends to mean for the growth rate of cells. In real, actual living cells, there is a temperature optimum for that cell to survive and for that cell line to multiply. And when you exceed that temperature, protein denaturation can take place. This is the idea behind the evolution of fevers in organisms like ourselves. Heat denaturation is an important step in things like PCR. You also need to know when you're doing this special gel electrophoresis technique, you can take detergent and coat the protein in negative charges so that the longer the protein, the longer the charges. And then basically this just means that you turn the bigger protein into something that interacts more with an electric field and the smaller ones interact less with it. The SDS is essentially, you can think of it almost like a soap that coats the protein and denatures it. That is what you need to know about protein denaturation on the MCAT. Visit mcatboss.com for more help.